The overall goal of this enhanced multiplexing method is to increase the number of protein samples that can be analyzed simultaneously, while decreasing sample error and instrument time.
This method can help answer key questions in the fields of aging, neurodegenerative disease, other age-related diseases, and cancer that are related to pathogenesis, progression, diagnosis, prognosis, and therapeutic targets. The main advantage of this technique is that a comprehensive snapshot of protein changes across many conditions can be generated.
Though this method can provide insight into Alzheimer's disease in mouse model, it can also be applied to tissue samples from patients with Alzheimer or a range of other disorders.
Generally, individuals new to this method will find it challenging because there are several samples to handle simultaneously in a short time frame.
We first had the idea for this method when we recognized that acetylation could be combined with isobaric tagging for protein nitration, which motivated us to make the technique work globally for all peptides.
This study will analyze brain, heart, and liver tissues from an Alzheimer's disease mouse model and wild-type controls. Transfer 60 to 90 milligrams of each tissue sample to a Liqing tube, and add 500 microliters of PBS with 8 molar urea. Homogenize the samples using a mechanical homogenizer. Remove the tissue homogenate from the Liqing tube and transfer to a microcentrifuge tube. Rinse the Liqing tube with 100 to 500 microliters of PBS with 8 molar urea, and combine the rinsed solution with the tissue homogenate. Centrifuge the tissue homogenate for 15 minutes.